Okay, so hello, good morning, and welcome to part 3 of Barcoding Bioinformatics. This is the final part in our series, so hopefully you've enjoyed watching, or if this is your first time, you can go back and see. I'm Jason Williams from the DNA Learning Center at Cold Spring Harbor Laboratory. And to get started with our final installment here, I'm going to go ahead and start sharing my screen. Got some slides for us. And going to introduce the topic and give some reminders, and then we'll get into today's lesson. So again, this is the DNALC live channel, and we're really happy to do this. This is the end of a very full week of activities, and we've got another whole full week coming up. It is an experiment. We really want your feedback, so please come to our website, share your comments on this video, come to our social media page, all those good things. Let us know how we're doing. The goal of this DNAC Live project is to really continue learning at home with molecular biology, genetics, and bioinformatics, to give some demos, whether it's in the lab or as we're going to do today in the computer, and for many different age levels, group school levels. We also have interviews with scientists, help for teachers, and a whole bunch of activities. So make sure, however you found us, that you go to dnalc.chl.edu and check out the live page. That's the best place to see what's coming up and what's planned. And on the social media, let us know again what you're thinking. Okay, so this is our final part in barcoding bioinformatics. I'm working on a series for next week, and we'll be announcing and posting more information on that soon. The audience for this course is if you've taken or are currently enrolled in AP Biology in the US, or introductory undergraduate biology. That'll give you the best position to kind of understand where we're coming from. If you haven't taken those courses, you may find some of the topics that you might need a little bit of refreshment on. But if you have taken those courses, that's what we're aiming this at. This is the last session. We're going to be somewhere between 45, 60 minutes. There are portions that are completely follow along with our tool, DNA Subway. We'll talk about that. And if you go to the DNALC live page I mentioned a few moments ago, you'll also be able to download the slides and the packet. And there's also an instructor's edition, so you can let us know if you're an instructor and you want that as well. The course has some learning goals, and we're going to return to these at the end, but just to state them for the last time, we want to learn how DNA can be used to identify unknown organisms, understand how the sequence quality comes into play there, use some software tools like BLAST to compare unknown sequences, and then today, compare DNA sequences using some phylogenetic methods. And the BLAST and also the understanding of trees is important for AP Biology, and the BLAST is in particular tested on AP Bio Lab 3. We're using DNA Subway, and we've mentioned this. If you Google it or if you haven't bookmarked it yet, you can find it just doing a Google search or going to cyverse.org. You can sign up for a free account. I encourage you to use the opportunity to sign up for that account because your work will be saved. You can use it without an account this time, but go ahead and sign up for an account when you have a chance. I believe next week's Bioinformatics, the next couple of weeks, next series will require an account, so you might as well do it now. Okay, so today we're going to focus on sequence alignments in phylogenetics. Let's recap our experimental data set and review what we covered last time from BLAST. And then also we're going to introduce two new topics. These are pretty extensive topics, not the easiest, but we will at least provide an introduction, and that's where downloading some of the packets that we posted gives you even more in-depth explanation and prepares you for that. As for a recap, you remember that the goal is that we have some DNA from unknown mosquitoes. We're looking at three particular genuses. And the problem as a taxonomist, let's say, or somebody who's working in public health sector is that as immature larvae, they can be fairly difficult, even to an expert, to distinguish them. As the adult, they have morphological features that are pretty distinguishable, but as the larvae, you may not be able to tell. And so identification, whether you're doing that for public health and deciding what form of treatment you might want to apply to bodies of water, or whether new species that haven't been seen in one place or another are moving, which is actually a problem. As we have a changing climate, species are going to move around, and therefore the diseases that they're vectors for may actually appear in new places. So we'd like to know that way in advance. So that's what we're doing. And the technology of the technique is this DNA barcoding. We are doing a bioinformatics laboratory here, so we're not covering the first few steps in a lot of detail, but once you sample your organism and extract your DNA, you can amplify the barcode via PCR and also do the bioinformatics bit. And we have another series on DNALC Live that is going into all of those steps in detail. So check the website, and if you want to learn more about that, please go ahead and take a look at those. Okay, so one of the things that we did last time was we used this technique, software technique called BLAST, in order to take an unknown DNA sequence. Once we had done some quality control, we talked about how DNA sequence is generated. Well, once you have that, you can actually go ahead and use the software to determine what is this unknown sequence. BLAST, as we said, is an algorithm that searches a database of sequences. We mentioned that NCBI, the National Center for Biotechnology Information at the National Library of Medicine, is one of the big repositories, one of the big databases for this sequence. And so we can search using the BLAST algorithm. The metaphor that you might use is Google for DNA. It's a similar idea, although technology is a little bit different. It's actually older, but it's what we use to search for DNA. And as I mentioned, this is a popular interface that we used both at NCBI, but then also DNA Subway, because BLAST is a software tool and you can use it in many different forms. When you're doing BLAST, a little bit of terminology, just a little bit more, is that you have a query sequence. So that's the piece of DNA that you started out with. It's unknown to you. And you're asking the question, can I find something in the database about which there's a lot more information? And so you've got to search the database. Now, this brings up a question which I'm prepared to discuss in a little bit more detail today, which is a really important question. Where in the DNA should we look for the quote unquote barcode? One of the things that happens even now as people live in the world of CSI films and forensics and things like that, is that when there's a question, when something is going on, let's say there's a crime, there is some kind of case, and people say, well, if you want to find out what happens, look at the DNA. But humans in particular have 3 billion bases of DNA. And so it's not at all clear where you should look when you are talking about looking at the DNA. What does that mean? And in fact, confounding that, and this is true of humans, but it's true of a lot of different species, is that whenever you're looking at a species, we're similar. And in the case of humans, we're 99% identical when we look at our DNA, in fact, greater than 99%. Now, when you look at us morphologically, we all look fairly different, right? Except for identical twins. And even once you know identical twins, people who know identical twins well probably can tell them apart. I've had that experience. You can tell them apart pretty well. But humans share 99% of their DNA, that greater than 99%. That's not apparent necessarily looking on the surface, but it is actually really the truth and something we have to think about. Now, to just sort of look at that or think about that, if you think about these two $10 bills, I can tell you that one of them is counterfeit. So if you look at them, they look pretty similar, right? Just looking at them, I would say they're 99% similar, given that, I know that they can't be 100% similar, otherwise one wouldn't be counterfeit. Now, I take this from a website, it's credited. I could not find any difference that made me feel I knew that one of them was counterfeit. Normally, you need touch or you'd be able to examine it more closely. But according to the website, the bottom one is a counterfeit. So that's a pretty good forgery. But it kind of captures, the situation we're in. When things look the same nearly everywhere you look, you need to know where to look in order to find that one detail that will tell things apart. Because everywhere we look, that's the message. Most genes that we would look at, if I look at here, I just took some tubulin, most genes, most things that we look at they're going to look exactly identical, no matter where we look. So we actually need to carefully select a region or a location, and we're going to use the word locus in the genome, that is going to have some distinguishing characteristics. It turns out that the gene that we're looking for when we're talking about animals in this slide deck, I'm only talking about animals, is cytochrome C oxidase, the subunit 1, which is part of this really fantastic looking protein structure that we have here on the left-hand side. And it's actually a mitochondrial gene. So if you were to look at the mitochondria on the right-hand side as a map, it's this one area of the mitochondrial genome that codes for a protein that is the distinguishing barcode for animals. And in plants, there is a different set of genes that we look at, fungi, but for animals, we've chosen CO1, and we'll talk a little bit more about why we chose that particular gene. So here are some factors that come into play when thinking about what to choose as the piece of DNA that you want to look at. What is the barcoding locus? There are many different criteria. I'm just going to choose 3 of them to focus on, but there are other potential ones that you might also consider, but let's just focus for these 3. I'm going to call them universality. I'm going to call them the second one discrimination, and the final one robustness. So the first one is universality. And one of the reasons why we use DNA barcoding and why it's such a successful and useful technique is that we can, take 100 different samples from potentially 100 different animals in our case, or whatever species you're barcoding, and we can just go ahead and amplify that DNA through the process called PCR. I haven't covered PCR here, but essentially it relies on being able to anticipate that a piece of DNA in that genome that you are sampling exists. So you have to choose a piece of DNA that nearly every species has. If you choose a gene that only a few species has, and then half the species don't have them, then when you try to barcode it, that half won't work. So a little illustration, of course, if you were to decide that you want to categorize candy bars based upon what type of nuts they have, well, that could work because, Snickers have nuts. If it's regular Snickers, it's peanuts. But there's also Snickers almonds, so that's okay. It's a different type of nut. And then you have peanut brittle. Well, they have nuts too, but how will you classify all the candy bars that don't have nuts? Now, Mounds has coconuts, and coconuts aren't really nuts. They're drupes, botanically speaking. So you need to, be careful about what you select, what your distinguishing characteristic is going to be. If we want to make it more biological, on the left is something called a dental formula. This is a great way to characterize a species, especially let's say looking at a skeleton. On the right-hand side, I'm explaining it just a little bit with a photo, but essentially, depending on whether your animal has incisors or cuspids or, canine teeth, molars, premolars, all these different types of teeth and how many, there's actually a dental formula which specifies them. And it's a really nice way, if you're just looking at a skull, absent some other features, right? The sheep and the dog skull look fairly similar, at least more similar than a human and a dog would. And so by looking at the teeth, wow, that's a nice way to be able to tell what you're looking at. But not all animals have teeth. And so this would work for the animals that do have teeth, but most animals don't have teeth, especially if you think about like insects, which are most animals. So we need to choose something that's universal. Next one is discrimination. So once we've chose something that we think everybody has, for the most part at least, you need to choose a characteristic that is going to be different enough that you can tell the difference between different people. And let's use that analogy of people. And one of the characteristics, if we wanted to tell people apart, one characteristic we could try is hair, right? Most people have hair, even if it's very short like mine. And you could say, well, I want to be able to tell people apart by their color, the color of their hair. And that could work to a certain extent. But you also run into trouble because the hair colors that we have in the human species are fairly limited. And also, they are very, very tightly confined to different populations of people. So for example, looking at this chart, which actually I believe comes from L'Oreal, who is interested in hair color. If you look, let's say in Asia, and we'll stick to Asia just for one moment. By and large, genetically, most Asians are going to have hair that's very, very dark, black hair color. So in Asia, if I were to go to a country, let's say Japan, and let's just assume only natural hair colors we're talking about, giving somebody the information that someone had black hair isn't very informative. It doesn't provide for you any discrimination because nearly everybody has black hair. So it's not the best characteristic. On a global scale, okay, if I wanted to tell a population of Europeans apart from a population of Asians, okay, there I might be able to have a little bit of discrimination in terms of that characteristic. But hair color might not be the best way that we could tell people apart. Especially if we're looking within, some geographically defined populations where there might not be enough variation to tell. So let's bring this back to the level of DNA. When we are looking at DNA sequence and when we choose a DNA sequence, here I have something called a multiple alignment, which is what we're going to focus on in just a few minutes. And I have different species. On the left-hand side, you see them named species 1 through 6. And this is a piece of DNA that exists in each one of their genomes. So they all have this same gene, let's imagine that this is. But for DNA barcoding, this is not a very good place to look because they all have just really the same DNA. So there's absolutely no way to discriminate, no way to tell the difference between species 1, species 2, and species 3. But if you notice my title, it says discrimination, but not too much. So what if we looked at a different region of the genome? So here is a different region of the genome. And in this case, there's almost no similarity between these different species. And okay, well, we can really tell them apart, but it actually is not very good for barcoding. The problem is that really with this small amount, almost non-existent amount of similarity between these species, I'm really, it really could be that these aren't the same gene that we're looking at in terms of, that they have a common homology. So it may not be that we're looking at the same thing and comparing oranges to oranges or apples to apples. And in addition, this is going to make it very, very hard to do PCR because in PCR, we design primers in order to amplify the DNA. And if every species would require a different primer, then we're not really doing DNA barcoding. We're not really getting what we're looking for in this method. So what you actually want is somewhere around 70% conservation. In other words, you want to be looking at a homologous gene where most of the gene is the same, and that allows you to have confidence that you're looking at the same locus for everybody, for all of the species. But you want there to be some differences sprinkled in there, and the differences you can see are mostly in areas where it's not highlighted. So you can see around, let's say this is nucleotide, or we'd say residue 15, when we're talking about alignment, we use that term. It's half of the species have a C and the other half have a T. So 3 C's here and then 3 T's there, and so on and so forth. So there are differences that eventually could allow us to sort and even make some inferences about one species being more closely related to another because they share more of those changes. So we want discrimination, but not too much. The final thing is fairly simple. I can't really show it in any good way. The best way I could show it is actually to say, look at a gel. What I mean by robustness is that when we are doing PCR, frankly, it just doesn't always work all the time. And if you've been fortunate enough to do PCR many, many times, hopefully many of them work, but you'll often have some PCRs that don't work very well. And there can be many different reasons why a particular region of DNA can be difficult to amplify. For example, if it is rich in G's and C's, those have a higher melting point, those triple hydrogen bonds for a GC bond. There can be that the DNA has what we call secondary structure, where the DNA tends to clump or loop, and so PCR may be less efficient. So once you've found a locus of DNA that meets the quality, that it's universal, nearly everything has it, and has the right amount of discrimination, you actually need to test it in the lab and see, does it work as a PCR experiment fairly well? You want something that works 90% of the time or more, perhaps. Okay, so those are the things that we're looking for, and I just wanted to summarize that. And then there's a nicer picture of this one area of this protein, this subunit 1. By the way, since the cytochrome oxidase C, I didn't mention it, but it's actually the last step in the electron transport chain. So it makes sense that nearly all animals have this, because we all respire, and so we all undergo those metabolic processes. It's a gene that's there. It doesn't change. It changes, but not too much. So it really is in that sweet spot, and it has, successfully become the major gene for DNA barcoding in animals. And there are other ones that you could use as well, but we're focused on this one. Okay, so what I want to do now, after that bit of explanation, is go take a look at our project and see where we're at and see where we're going next, because we have a couple of experiments to do. And I do want to use the term experiment. We've gone last time and introduced our controls and all those different things. In fact, here we are. We have our materials. This is our experimental design. Remember, bioinformatics, you are doing an experiment. Materials, we have the DNA from the unknown mosquito samples. We have, and we are obtaining through BLAST DNA from some known samples. Our hypothesis is that we can use these computational methods using certain parameters in order to infer what the species is. And we have a whole set of controls. We have sensitivity controls, which are our sequence quality and our BLAST parameters that we covered in the last couple of episodes. And we have outgroup sequences, which are non-mosquito DNA. Those are negative controls, and also known samples. Those are positive controls. And those are going to matter to us in the experiments that we're going to do today. So first, we need to introduce multiple alignment, but let's go ahead and open DNA Subway and just take a look. I am going to share that. Okay, and I've already logged in. Let me just go to the home. So hopefully you are on DNA Subway. Let me also open this, since I have not shown it in this episode yet. If you go to DNALC.org and you find the live site, I want to make sure you know where the handouts are. So here we are right on the front page, because this is current, but if you go to DNALC Live, you can see all the events, past, present, future, I guess. And we're in Barcoding Bioinformatics. And under the Resources tab, I really encourage you to take a look at that. Both the worksheet for today and also the handout, because it's got a lot more information. There's a lot more even than I can cover. And if you haven't started the experiment and gotten up to the step where I'm at, you can go back there and follow along. You can also watch the other videos which are posted to our site. Okay, so if I go to, I've logged into DNA Subway. If I go to my projects, I'm going to find the project that I started for this webinar. In our sequence viewer, which we've covered many times, these are our raw, uncleaned, or unedited, unfiltered DNA sequences from these various unknown mosquitoes. Okay, we filtered them, we cleaned them, and we paired them up, and we ended up with consensus sequences. So we now have 4 actual samples of mosquito DNA, and we've really trimmed them to make sure that we've gotten rid of any low quality sequence that we're not confident in. Then in the next step, we went to the BLAST tool. And last week, we just did 1 or 2 of these BLAST searches, but the homework was to make sure you did them all, and I've done them. If I click on any one of them, let's click on mosquito 1. I get some results. If I click on that first result, this case, this is for, actually, this is from a species that is from, if it says bold there, I'm thinking it's from the Barcode of Life online database. So it's some kind of 80s species. That's a genus. I'm going to skip down to one of the ones that actually has probably a photo attached to it. So this is 80s Vexen. So we're getting some nice mosquitoes as our hits. We explained a little bit last time what the results here mean. So we have an alignment length, which is how much of our sequence matched a sequence that exists in the database. The bit score, I went into a little bit last time. but more important is the expectation value, the e-value. Lower is better. And so 0 means that there's, it's extremely improbable, basically impossible if it's 0, that this is a match due to random chance. You'll also see occasionally that there's some mismatches, but even with one mismatch, we're largely coming up here as this being some 80s, although there are a couple of culinae that are coming up as our blast hits. Now, one of the things that I also mentioned you could do, if you haven't done so far, is if you find a species, you can go ahead and select it and then click add hits to your project. So that may be important. And the next thing to notice here is we have this thing called reference data. So in reference data, we provide you with a list of species, and there are also some DNA data sets associated with those species. And we've tried to choose representatives of some of the major families. So when I click on common insects, these are some of the more important orders that are present. And so if I want to go ahead and rely on this or make use of these species, I'm just going to go ahead and click add reference data. And so later on, I can come back and examine them in what I'm about to do next, which is a multiple alignment. Okay, and the next thing I do, once I selected some data, if I want to continue my project, I can come along and say I want to upload or import data from other sources. There is a DNA Subway manual, which is linked at the bottom of the page, and you can see any of the features that I'm not going into just this moment. But the next thing we're going to do is we're going to select some data in order to start making some comparisons. So these are all of the data sets that we've accumulated over the course of our experiment. And remember, the user data, this one here, our mosquitoes are the unknowns. So these are our experimental data set. And then everything else can be thought of as some type of positive control, because we happen to, we believe what the label is, this really is a cockroach, this really is a dragonfly, this is a fruit fly from the Diptera species. So we should be able to believe that these are some data sets that we could use in a few moments. And what you have the opportunity to do, we're going to be building, and I'm going to introduce now the topic of multiple alignments, is you can choose and you can go back and reselect and re-choose and shuffle things around. So we're going to make use of that in just a moment, but let me go ahead and share this information on multiple alignments, and we'll come right back to this. So I'm going to go back to my slides. Okay, so this is the next major topic, multiple sequence alignment. And so if we want to compare the sequence features, and I'm talking, right now about DNA nucleotides, but I could actually be talking about amino acids, whatever type of sequence. The procedure is essentially, it sounds simple, but there's actually some complexities to it, is we need to line them up. So it's like this gigantic puzzle pieces. We need to get them all in some type of order in order to make comparisons. So I'm going to use an analogy, and again, I warn you, it's only analogy. It's not meant to perfectly recapture all of the concepts here, but it's enough for us to be able to have a little bit of discussion. So here are some sequences. These could be protein sequences because in proteins we have 20 or so amino acids, and so we use one-letter abbreviations for those amino acids. But if you look really closely, these are not protein sequences. These are actually strings of alphabet, actual language. And the first one, if you look carefully, is English. Although there's no spaces there, the first one is English. And if I give you a little bit of a clue, I can actually make an alignment. So we have some different languages here. We have English, which I said is the first row. The next one is Scots, which is very, very closely related to English. Although I've taken out things like the glottal stops, which are the apostrophes, and I've taken out diacritical marks where they've appeared. Then going out to some other languages that are closely related to English, which English is a Germanic language. So we have Dutch there, and then Afrikaans, and then German, and then Icelandic. So these are all languages which actually have a bit of relationships in common. And so the analogy is actually not a horrible one for how DNA sequence changes. And in fact, I use the same software that makes the alignment for DNA and protein sequences to go ahead and take these language sequences and put them out there to help us understand a little bit about multiple sequence alignment. So in this slide, what I've done, and this is what we also often do in a multiple sequence alignment, is we try to figure out how similar sequences are by finding out how many nucleotides or how many residues or amino acids do they have in common in the exact same position once you line them up. And you could see that in some cases, right, where there's a missing letter, then we have a dash, right? Or sometimes you need to infer that there were some missing letters and add some dashes so that things line up. And in this case, I've colored things if they share at least 60% identical. So in other words, if most of the letters in that row or in that column are the same, then it gets colored. And you could see that a word like water has, well, that's kind of my next slide. If we look that there's more information here, a word like water pretty much stays the same. And actually, if you are a linguist and you're going into an area where somebody speaks a language that you don't speak, and you're trying to decipher a new language, what you try to do is figure out, well, are there languages that we have a dictionary for that might be related to this language? And then you also start with the most common words, because it turns out that usually the words that are the most common are the words that change more infrequently. So a word like water or a word like child or mother, those stay pretty fixed, even in languages that were once the same language, but now have branched off maybe for hundreds or thousands of years. If you even look a little bit deeper, some of the words where even though the letters have changed, the sounds are still similar. So there's different ways, like the vat in the Icelandic kind of still retains that WA or VA kind of sound, right? And if you go further, you know, you can see the word the, you know, even though the letters have changed, the sounds are similar. And if you keep going with fish, right? So this is really a very good analogy with language. And it's a concept that we need to call upon when we're thinking about multiple sequence alignment. Now, the problem and the reason, again, for bioinformatics is that, you know, I just made this alignment for a few short, really a sentence in a few languages. But as it turns out, there's a bit of math, as there always is in a bioinformatician's life. And that is that if you want to actually line up many sequences, so maybe you have many, many different species, and then those sequences are some length. For DNA barcodes, we're often looking at somewhere between 400 to 600, maybe more or less, nucleotides. As you get longer, the amount of possible ways that you could line things up gets greater. And so it turns out to be computationally challenging as the problem gets bigger. And what that means, this equation, is that if you have two sequences, which are only 100 nucleotides long, there's 10 to the 77th, you know, possible ways that you could line them up. Now, that's a little bit misleading. It is still a big number, but this is slightly misleading because some of those alignments will be basically the same thing and trivial. But the key thing to take away from this is that you actually need software in order to do this well. So there is an algorithm. We're actually using muscle, but I thought I'd mention this one called Clustal. It's one of the first ones that really came out by Des Higgins, and he's at actually University of College in Dublin, and he is this is one of the first ones to come out to do this type of alignment, multiple sequence alignment. And it's an algorithm, so there's a couple of steps to it. That you have different sequences and you try to line them up not as a group. Like if you have 20 different sequences, you don't try to make the whole thing at once. You just align number 1 with number 2, number 3 with number 4. You make what we call pairwise alignment. So you try to simplify that. And then you form a matrix where you calculate the similarity between those different sequences. You make what we call a dendrogram, where you try to figure out which are the most closely related based upon that, and then you build that alignment out. So it's a fairly straightforward, but still, it can become a computationally intensive procedure, especially as the problem grows. And the other thing is it's also imperfect. So it tries to make this alignment. It has all sorts of scoring matrices and things like that, kind of like what we use in BLAST. But oftentimes you need a human being to come in later and clean up the alignment. So it's not perfect, and we're going to see that. So let's take a look and let's build our own. We're using a different algorithm, one called muscle, but many of the concepts are still the same. So we're going to go back to DNA Subway. And the first thing that we need to do is we need to choose which data set we want to go ahead and align. Now I've actually conceived this. Let me go back and share my slides. Oh, what we're going to do is I'll do it this way. I don't have to share my slides yet. I can stick to DNA Subway. I didn't want to get ahead of myself. I'm just going to go ahead and choose our data, user data. So we've got our horsefly and we've got all of our mosquitoes. And once since that's the only one I'm going to do for this second, I'm going to go ahead and say save selections. And once I've saved them, I can go ahead and click on muscle. And it's going to take a few seconds. You can kind of notice it didn't take more than like 5 seconds. And you'll notice that will change when we go back and choose different numbers, but that's going to be enough right now. So click on muscle. And I want to orient you as to what you're looking at. Now, the very first thing I hope you can appreciate is that we've got this little pattern here that kind of looks a little bit like a barcode. So what are we looking at? Well, the first thing just overall that you might want to sort of appreciate is that each one of these colored bars gives you each one of these colored bars is an indication that there is a difference in this particular species, and it's a difference that maybe is not shared by all the other species. That might become a little bit more apparent if I zoom in. So I'm going to click the plus button on the top. And at this level, I mean, the bars are a little bit, they're nicely colored. I can see them a little bit better. But if I click all the way in, what I start to really get is the actual nucleotide differences. So let's draw our attention up to the top, and then we're going to see a little bit more. The first thing that you see on the top is actually this row called sequence conservation. And this row can be thought of as a histogram. So when it's all the way at the top, it means it's at 100% conservation. What that means is that every species that's listed in that column has that nucleotide. So if I scroll down to something that says 40%, the histogram is lower, and that means of the consensus sequence, which I'm going to mention in a moment, only 40% are matched to that. The next thing that you see is a thing called sequence variation. And sequence variation tells you by color, if there is variation in that column, what variation do you see? So if you look in this particular column, we don't count the consensus sequence, so don't worry about that. But if you look at all the differences from the consensus sequence, it says, excluding the consensus sequence, what are your other options in this column? And since this is blue and red, it means the only other options are C's and T's. So in other words, the only other color that you'll find here, if it's not an A, is either going to be a C or a T. If you go to this column here, the only thing that you're going to find if you don't have a T, that's a consensus, is going to be an A. So that's the only other option that comes in that column. And the consensus sequence is the average. So it's the nucleotide that by frequency occurs most frequently in that column. Wherever you see the consensus sequence, if you don't see anything beneath it, that means that they're all the same. And if you do see something beneath it, it's a difference. So as we scroll through, let's go to area, like this area here has a high amount of conservation. It's nearly 100 for, let's say, this is 15 or so nucleotides. And then eventually you see that there's some differences here. You can also start to think, sort of look ahead, that mosquito 3 and mosquito 4 have a T here. Does that mean that those two are more closely related? Maybe. I don't want to go by one nucleotide. Mosquito 1 and the horsefly have an A here. Does that mean they're more closely related? No, you can't go by one single nucleotide. But the thing that you start to be able to imagine is that if we can find enough of these patterns and calculate them in a really nice way, we may be able to figure out that some species are more closely related. The next thing I want you to pay attention to, because it's really important before we make comparisons, is that as you get, sometimes at the beginning, but really also at the end, you get these areas where you have this really dark gray. And that dark gray here indicates, whoops, I clicked back when I didn't mean to. That dark gray on the alignment, am I in the right place? Let's see. That dark gray means that those sequences are missing some data. And so before we actually use this alignment in anything, we want to click Trim Alignment, just so that everything is lined up at the same exact place. Okay. So whether we click to the nucleotide level and scroll all the way down, now we can see things and in a little bit more detail. And the reason why it's very important to trim your alignment is because if you don't trim your alignment, the software later on will imagine that those missing pieces of data or DNA are actual real gaps in the DNA sequence. Like there's been massive deletions or massive mutations. Usually those gaps at the beginning and the end is just because that's where our sequencing ended. The final thing that you could see here is that there's a matrix that's computed called the sequence similarity. And if you click on that, it gives you an idea pairwise, but what I mean by that is by comparing any given two sequences, how much they have in common. So sequence 1 compared to sequence 2, this 85.82% identical, so on and so forth. So you get this, these matrices are calculated all the time. Okay, so that's a multiple alignment. And the software that we use to take this multiple alignment and actually determine what the relationship is the next set of software, which gives us our phylogenetic trees. So let's talk about that in the last 15 minutes or so that we have. Go back to my slides. Okay, so we're going to talk for this last section on phylogenetic trees. And this is a very complex topic, and it's something that a lot of people may find difficult, especially if they're not in this area of biology. But we can at least get some of the basic concepts down and make use of it. So many times in biology, if we're talking about tree relationships, maybe this is one of the everyday things that you could relate to, like a family tree, where you have a pedigree, you have, the mother, the father, the different generations. But this is not a phylogenetic tree. So in some ways, maybe the idea of tree is common to this pedigree, but it's not what we're talking about when we're talking about phylogenetics. So this is more close to what we're talking about. And the key takeaway, if you take away anything from what a phylogenetic tree is or from this discussion of it, is that a phylogenetic tree is a hypothesis. And it's a hypothesis about how species may be related. Almost always when we're dealing with phylogenetics, we're dealing with incomplete information. We don't really know how species might have been related, but we can gather evidence and we can make some inferences and we can make some hypotheses based upon those evidence. So here is another way of looking at a phylogenetic tree. The first one was trying to group some of the major kingdoms of life. Here's one where we can also think about a phylogenetic tree as telling us something about traits that we expect, or mapping traits onto a tree. The tree reflects those traits or those characteristics. So if you look at this tree at the very bottom here, we have this little tick mark that says vertebrates. And what you'd expect is that everything that you see from that point forward on the tree is something that has a backbone. If you look at the idea of lungs, right, you'd expect that things that come after that tick mark have lungs. So that's true. But things before that don't have lungs. You can even see that sometimes a trait like hair arises, but it arises only in one lineage. So mammals have hair. If that hair tick mark had been down here next to the amniotic egg, then everything else going forward would have hair, but that's not the case. All right. So when you're looking at a tree, or here's another example, you could imagine that if we have trees organized by what type of nuts they have again, you might get peanuts, right, for paydays and Reese's and Butterfingers, that they may come together. I'll come back to those words, branch and tax and tips, in a moment. So here's a phylogenetic tree that somebody did. I just wanted to make it clear that although we're using DNA sequence and you could use protein sequence, you can take any characteristic and make inferences about how things are related. So in this tree, people made it, tried to make inferences based upon things like the shape of the candy bar, or also the type of nuts or the type of flavoring that it contained. So these are all examples of ways that you could take trees, or that you could use them to really emphasize that it's about relationships. It's not that necessarily we're talking here about an evolutionary relationship like Crunch Bar came from Heath Bar or something like that. We're really thinking about what are the inferences of relationships, and those, and what are possible hypotheses that are supported by the data that you see. Now I want to come back, because you might have been distracted as I was by the candy, but I want to make sure that you get a little bit of the vocabulary as well. So when we talk about taxa, those are individual species that usually are right at the end of the trees, at the tips, those are the endpoints. Then you also have nodes, which are sort of branching points, and then you have different branches, which are the actual, anything that comes out of it after a node could be a particular branch. There's this and a handful of other terminology. Some of it gets used more strictly, some of it less strictly, but it's at least good to know some of these terms. So now what we can do, because remember, when we did BLAST last week, one of the things I told you is that the BLAST result alone does not tell you whether you have identified a species or not. And I could make a similar slide here, but the idea of, if we have BLAST evidence and also we have phylogenetic evidence of what species we're looking at, while I can't tell you that things are exactly, that you've reached an infallible conclusion, we can at least come to some kind of conclusion that we have a lot of evidence for a particular inference. These are types of experiments that we could do, and I'm going to explain what they mean in just a little bit. But before I get into doing those possible experiments, I don't know that we'll do all of them today, but they're set up for you in one way or another in the packet. I want to take you through it, but I also want to point out that when we're making phylogenetic trees, we also run up against a lot of computationally intensive things and some math, because as it turns out, when we want to infer these relationships, we want to try to make these relationships, there are many, many different ones that you could make. And if you have many species that you're trying to make a tree with, by the time that you get to 10 species, you're talking about literally millions of possible combinations. And if you're trying to make a tree for 50 species, it's just an outrageous combinatorial problem. And so we use all sorts of algorithms in order to make the tree. I'm going to talk about those because there's no one best method. But before I talk about them, let's do our first experiment. Our first experiment is we're going to take our unknown mosquitoes in the out group. which is that horsefly, and we're going to make a neighbor joining tree. So I want to at least get to that hands-on portion. And then we'll come back and talk a little bit more about the methods. So back in select data, make sure you feel comfortable with this so you can do the ones that we don't get to. I selected the horsefly and I selected the other 4 mosquitoes. So I'm not going to make any changes. I'm happy with that. And then in the mussel, I've got my sequences there, and then very importantly, I've trimmed them. Now the final thing I can do is use this software called Phylop, and I'm going to use a method called NJ, or neighbor joining. I'll explain that in a moment. And when I click on that and open it, I am going to come up with this tree. Okay, so this is a tree, not the only possible tree, and we need to interpret a little bit, but we can already see some relationships. In this tree, we've got mosquito 1, 2, 3, and 4. We've got an outgroup, and actually on the left-hand side, we should select our outgroup as being the horsefly. And outgroup just means that it's a species that we know is not related or not closely related to the other ones. So when we do it like this, we see mosquito 1 and mosquito 2 coming out in a particular branch. And if we expand that back 1 node, we see mosquito 4 as being the next closely related, and then we have mosquito 3. And then finally, all of the mosquitoes are on 1 branch, and then the horsefly is on a separate branch by itself. So that's good. Let's go back just for a moment and see a little bit more about what we're talking about. Okay, so this neighbor joining method, this is one algorithm for drawing a tree, because remember, the computational problem is that when we have a number of sequences, so in our case, right, we have at least 5 sequences, and we're going to add more in a moment when we add some of the other insects. Even when we add the insects, right, we're going to have at some point more than 10 sequences. So we have millions of potential trees we could draw. So how do we evaluate them? Well, one algorithm called neighbor joining is what we call distance-based method. It works very similar to that multiple alignment where we ask what's the similarity between 2 sequences. A matrix is built, and the distances between those 2 sequences are calculated. And the distance is related to the number of mismatches between the 2 sequences, which we saw in our multiple alignment. The reason we line things up is so we can tell where do we have perfect matches and where do we have mismatches. And then the other cool thing that we get is we get this thing called the Bootstrap Value. And the Bootstrap Value tells us, this is the way that we evaluate neighbor joining tree. If I were to randomly sample columns from my multiple alignment, and in this case of DNA subway, we randomly do that 100 times, how often would that same exact tree appear? So matrix is created just like the previous, but basically it's asking us the question, if we were to do an experiment where there's a bit of randomness involved, and you have to appreciate that. So when we're building a tree, if there are millions of possible trees that we could construct from the data, we can't build all of them and then evaluate them. So we're picking some, and we're asking the question, if I pick them out of a hat and look at their shape, and if I do it 100 times or if I do it 1,000 times, if I still see the same shapes appearing no matter how many times I sample them, then I can have a little bit of confidence that that is maybe something close to correct, right? So that's a key insight that you want to remember. Back to sharing this, the slides for 1 second, we'll go take a look. What we generally consider is that if we have a value of 70 or above, that's what we call plausible, and 95 or above is what we call highly supported. So if I go back to Subway, let's choose this. That relationships, all of these are above 70%. So it's pretty strong evidence that mosquito 1 and 2 are more closely related to each other than they are to mosquito 4. And that mosquito 4 is more closely related to mosquito 1 and 2 than it is to 3. Now, that doesn't mean that, you know, that's 100% correct, even the 100, right? but it gives you an idea of what the likely inference is. And then there's really, really strong support that these mosquitoes are more closely related to each other than they are to horsefly. Now, let's go. I gave you a list of experiments that are possible, and I encourage you to look at the packet. Before I go to maximum likelihood, which is the last thing I'll have time for, let's add some of those other species there and see what we get. So I'm going to go back to select data. And this time I am going to add the other insects. And I'll also add some of my blast hits. So I'll add 1 Aedes. Let's do this one that we have a species, 1 Aedes, and I'll add 1 Culex, because those are really, in fact, actually, let's do, before I do that, let me just go add Aedes and Culex, keep it simple and save it. I need to go back and do the muscle alignment. Okay, I need to trim. And just give that a second. Okay, and then I need to go ahead and build my neighbor joining tree. Okay, now when I do that, okay, we got to go back and select the horsefly as the outgroup. Okay, so now I have mosquito 1 and mosquito 2 coming back as a group that's well supported. And I have, of course, Aedes as being that. So does that mean that perhaps mosquito 1 and mosquito 2 are Aedes? And if I look at number 4, are they as 1, 2, and 4 also perhaps Aedes? And then for mosquito 3, I have something that's potentially Culex, right? So I could go back, do this again, but maybe add some more to it. So let's go ahead and add all of my Aedes and Culex. Okay, these are all the blast hits I retain. And I'm not going to add all the insects, but I'll add a fruit fly, which is also an outgroup. I can add a cockroach, maybe a butterfly. Okay, I'm going to save it. And you can do lots of combinations. I encourage you to look at the packet. I got to do those steps again where I choose muscle. Trim my alignment so everything is nicely aligned. And you can also start to see patterns, especially where you have blast hits, where we know the blast hits are already similar, where, you know, clearly this is a nice pattern emerging. And then if I go ahead and build my tree, okay, now I've got something that's more complex, and more complex than we could spend a lot of time on. But as you look, okay, we have the horsefly as the outgroup, and you have the horsefly. Then you have, and it's interesting here, we have the cockroach, we have the monarch butterfly, we've got the fruit fly, and it's interesting that our horse are actually here, the most distantly related are not the horsefly, because that's a type of fly, and the fruit fly. So we should actually choose a different outgroup. It could be the butterfly, although that's not truly a fly. And actually in the packet, it goes into that question. There we go. So we have horsefly and fruit fly coming out as a group. And if you look at it, this whole branch after this 100 could be considered the fly, right? The order Diptera. And if we look at things like our mosquito 2, we have it coming as an Aedes here, but mosquito 1 as more closely related to a different Aedes. And then we have mosquito 4 coming out here, and is it with the Culex, mosquito 4 and mosquito number 3? That's a lot of complexity to think about. And so the packet will go into breaking down this mystery. But I have one more thing to show you, and then we're just about out of time. What I'm going to do is just simplify this a little bit and point you in the right direction. I'm going to go back to my data, and I'm going to get rid of the other insects for just a moment. And I'm going to keep myself to maybe 1 Aedes and 1 Culex species, just to keep it a little bit simpler, because I'm going to save the selections there. I just want to talk about that last method. I'm going to trim, so you kind of know how this works. And then once you trim, well, I got to click trim. It was loading before. I'm going to also use this other method called maximum likelihood. And when I click on that, I also get a tree. And the question that you need to ask yourself is, does this tree show you the same things as the other? I've got my outgroup of horsefly. I've got Culex coming out with number 3. I've got 80s coming out with number 1. And 80s 1 and 2 are a group. The next closest is number 4, and then 3 is by itself. So are these trees similar? Well, the last thing I'll talk about and then move to wrap this up. Okay, these are some of the possible experiments, and I encourage you to go ahead and try them, is that this last method, which is a different algorithm than the neighbor joining, also tries to take into account about patterns of how nucleotides and amino acids change. It turns out that not every mutation is equally probable, and that the tree with the overall, what we call likelihood, which is a mathematical score, they don't show you, but you usually report it in negative log likelihood, is what's accepted as the best estimate of the relationship between the sequences. And the length of the branches tells you a little bit about the distance. So very short branches that separate sequences are lower confidence, and the relationships are less certain, while longer branches are better supported. So it's a lot to look at, but I encourage you to take a look at the packet. It goes into more detail than we could in a 1-hour episode. So what were the goals? And this is sort of wrapping us up. Well, we learned that you can use DNA in order to identify an unknown organism, and you need to choose the right barcode region for that to happen. We learned that even once you have your DNA sequence, you need to assess its quality and do some quality control before you use it in your experiment. And then we also learned that there are algorithms and software methods like BLAST and also Phylogenetics that help us to answer some of those questions. And I encourage you again to go to the handouts. So also, the other thing is, you can go back to DNALC Live and review the other videos that we have posted and leave us some comments and maybe have questions about going through this yourself. That was certainly the hardest episode because there's a lot of content to go through. So you might want to watch it again, and you might want to post some comments. I don't see any in the chat right now, any questions for me, but hopefully you get some benefit from looking at it. I can tell you that most people who don't study phylogenetics, even though they may be biologists, it's something that's a little bit difficult to master, but with a little bit of effort and some of the guidance and what we provide you in the packets, you can start to tease out how it can be useful in order to build strong evidence that you've identified an unknown species. So hope that was useful. And since I don't see any questions, I'm going to hope that you enjoyed the series and look forward to the next one. Thanks.